uh, uh, Dr. Sidhu.
Yes, I'm trying to share the screen, but it says I can't until...
Wait. If I've been given the authority, then I should be able to do it. Let's see. Right.
Go ahead, Professor Sidhu. Go ahead. You can share now.
Oh, good.
Okay. People see my slides now?
Yes, we can. You want to go full? You want to go full screen, Dr. Sidhu?
It should be full screen. You don't see full screen.
Everybody see full screen? Let's see if we got anything on the Q&A. All right, go for it.
Okay. Do you see the full screen or not?
I see full screen.
Okay,
Yeah,
good.
I see full screen too.
Okay, great. Yeah, thanks for the invitation. It's a different group for me, but I think everybody's doing stuff a lot differently these days, which is a good thing in some sense. We're reinvestigating our kind of role in science, and I really enjoyed the previous talk because it's clear to me now that what we're doing is kind of the opposite of what I think a lot of the aerospace industry does. We're interested in inner space, which is anything smaller than you can see. That's how we look at proteins and cells. While you guys work on millions of kilometers, we work on the nanometer scale. So that's what our goal is. And what I'm going to talk about is our efforts at applying our knowledge of biology and protein structure function to respond to the COVID-19 emergency in real time, which, as you know, science is not always, that's not what we normally do. We have a very methodical approach, which we can and should employ. But this has been really an interesting time for me, and working on a scale and time scale that's different from what we're used to. And I thought I was invited to do this, and it was very encouraging for me because in a way the group I'm talking to is very different, but there's something we share in common. I'm a protein engineer. So for the past couple of decades, we've been applying engineering principles to proteins. Do we understand proteins well enough? And the biophysical rules to engineer proteins in real time. And this has been a great opportunity to apply that, but we've been doing this for a long time. And I'm actually glad to be virtually in California. Before I came back to Toronto, I'm a Canadian. About 12 years ago, I spent almost 12 years at Genentech in South San Francisco, so Northern California. So I'm very familiar with the state and kind of the innovation culture there. So it's, and what I've been doing in Toronto, which I'll tell you about, is trying to bring kind of the high throughput industrial approach of biotherapeutics, which I learned at Genentech, but applying it on a really bigger scale and kind of to unmet medical need, which is what I think academics does or should excel at. So if you can see my cursor, I'm actually located at a center we call the Toronto Recombinant Antibody Center, which is in another center called the Donnelly Center, which is all in the University of Toronto. And so my center is basically my lab, and associated collaborators, both in academics and industry, and that's important for us, are trying to apply very basic research to real world problems. I actually became an expert on COVID-19. I can remember the date, March 1st. I was in Rio with my family. My wife's from Brazil. We flew back. We'd applied for a grant in February. When we came back, I got the good and bad news that we'd actually got the grant. So this was science in real time. So I'm like, we said we're going to do something. Now I guess we should. So, you know, I'm basically learning as we go, but I think that's kind of the lesson of these last couple of months. There are no rules. accept what works and who can contribute. So around March 1st, in fact, we started working on this problem. Of course, I know more than I'm implying, but COVID-19 was new to everybody. And I'm not a virologist. What I am is a protein engineer. But the motto of our center actually comes from a great group from Southern California. We like to say the Toronto Recombinant Antibody Center is all about medicine with attitude. And I hope to explain what we mean by that. Our goal is to develop therapies using the most cutting edge technology, and particularly in areas where we may not be the experts. There's nothing better than having an impact where you don't belong. I think that's a lesson we're learning here, that we need new attitudes and new methods. And in fact, Dr. Thangavalu had some great quotes. I'm gonna throw out our mission statement. It comes from the great American James Brown, who once said, you gotta use what you got to get what you want. And we really run our center with that mentality. What we're trying to do is, what do we know well, which is proteins? What are we trying to achieve? New therapies. Let's just get at it and see what we can do. So with that attitude, really, we looked at COVID-19 as an opportunity to apply engineering principles in real time to a problem that's evolving as we speak. And I want to stress one thing. We don't know everything there is to know about this disease, but we know a lot. I think that's what we've been trying to stress in our communications with the general public. We are by no means blind in this process. Decades of research going back to SARS, Ebola, HIV, polio, the flu, and in fact, SARS is a relative of this. So we know a lot. And I'm showing here a very simplistic, but again, real picture of what the virus does, what the immune system does, and therefore what we would like to do. Because on the plus side, this is a bad virus, but it's not, let's say, a global destroying level virus. It has limited lethality, an acute life cycle, and I'll explain why that's important. So there are ways we can tackle this virus in real time. The first thing a virus does, any virus, of course, is get into the cells. And once it's in the cell, it starts to replicate. And we know from HIV and other virus disease, we can interfere at any of these processes. So the virus has many weak points.
Excuse me, Professor.
Yeah.
I think your slide sharing is stopped. Could you reshare it again?
Oh, really? Sorry about that. Let me see. Okay. Go back. Do you see the slides again?
Yes, yes.
Okay, good. So this is the virus life cycle in a simplistic view, but it does bring up, if we can stop the virus getting into cells, we can prevent it from ever replicating. If we can stop the virus from replicating, that's another way. Finally, if we can stop exit, that's the third way. And we're working on all of those, but for today, I'm going to talk about our most advanced work, which is preventing viral entry. And I would say if we can achieve it, probably the most powerful approach. If the virus can't get into a cell, it simply can't do anything and will be cleared very rapidly. And with COVID-19, I must stress, its life cycle is on the order of 2 to 3 weeks. If you don't succumb to the virus, you do completely recover. And of course, as a lot of research has shown and was already known, one of the key means by which the body fights the virus is, of course, antibodies, shown in green. And the most simple, effective means it does so is antibodies bind to these spikes, which then prevent the virus from interacting with the host. And it really is that simple. If the virus cannot interact with the host cell, it can't enter the host cell. And more evidence is emerging that this is a virus that is highly susceptible to the natural immune response. That's why it's not high lethality. I know that's not comfort for people who are suffering with it, but it is something that perhaps 98% of people will recover from without any intervention. And we now know from many research groups that recovery correlates with large titers of neutralizing antibodies. And these are important things to know because we can do something about that. If recovery is induced by neutralizing antibodies, we can try to make vaccines. That's what people are doing, not my group. My group is thinking even simpler than that. Why don't we cut to the chase and make our own antibodies that we can put into the immune system, or sorry, into the bloodstream, and achieve what nature, our body, is trying to do directly. And engineering, as I'll show you, allows to do that. Because I'll emphasize this again, we know this virus intimately. This is a 3D representation, but it's remarkably accurate. We have cryo-EM structures of the spike protein. We have cryo-EM structures of the virus. I'm not getting into the details of inside the virus, but we know what is inside the virus. So we literally know what this looks like, what it's trying to do, and what then we have to prevent it from doing. And on the right here, I'm showing, angstrom level picture of the key spike protein. This is the protein that binds to the host cell and mediates entry and all the bad things that follow. From a lot of work from SARS and now with SARS-2, remember this is 80% identical to the original SARS. So my thinking, and we're proving it, everything we learned from that virus can immediately be translated here. We not only know the structure of the viral spike protein, we know that the very tip of it has an interaction domain, or the receptor binding domain, which binds to ACE2, a human cell surface receptor. And we know from studies with SARS, studies with other viruses, and crucially, with data emerging from recovered patients, that blocking this site is a great means of blocking the virus, period. And if you think about it, to me, that's mind-blowing. Within months, we have learned what, even with HIV, took decades, or previously we didn't know at all. What is the crucial Achilles heel of this virus? It clearly is the tip of the spike protein, which we have structures of at single angstrom resolution. So that's the idea of inner space. We can see this virus as clearly as we can see anything with the telescope. So knowing this, we ask the question, this is what we want to tackle. Natural antibodies do this. Can we do this with our methods? Our group is an antibody group. The immune system works this way. Natural antibodies, an injection here of antigen. This could be the SARS virus. Within weeks, you get an antibody response. And what I'm showing here is how we've subverted that in the past, or used it to develop what we call monoclonal antibodies. These are purified single antibodies that do what we want to do. But this is exactly the natural immune system. Of course, the problem with COVID-19 is it sets up a race with our immune system. If it can more rapidly outpace the immune system through replication, it can cause severe disease. And if it gets to the point where it overwhelms the immune system, it obviously leads to mortality. But we do know that antibodies are good. We know what they need to do. And crucially, the immune system has given us more than enough information about how to stop this virus to then attempt to do so. So natural antibodies are good, but synthetic antibodies are better, as I'll hope to convince you, because these are engineered molecules. These are molecules we make literally in a factory, a fermentation vessel, that do absolutely what we want to do. And I should say we've leaned on decades of work from Genentech and others on how do we take antibodies that are undefined and natural, shown in red, and through engineering methods, convert them on the bottom. This is from Presta and Carter, the paradigm for how to make therapeutic antibodies, which have already revolutionized cancer and other therapies, and I hope will revolutionize infectious disease in the upcoming decade. We already know this works. 8 of the top 10 drugs are now antibodies. More than half of the drugs in clinic are antibodies. So precision engineering of these molecules is not only feasible, it's done all the time. My group has done it, others have done it. And that's a huge advantage. We're not going into this blind. And showing this antibody here, it's of course microscopic, but it is a large molecule. It's 150 kilodaltons, very large protein. But I must stress, we know the angstrom level detail of every atom in that protein, what the different domains do, what we need to engineer. So we have precision at an engineering level of antibody molecules now. So we don't need mice, we don't need the natural immune system. We can use synthetic methods to achieve this. And the way my group has done this is a data heavy slide. I can send people to literature that highlights how this works. But basically, a Nobel Prize, I think the year before this year, was given for the idea of phage display, which takes the natural immune system, puts it in a tube, and it's, in this case, it's virus fighting virus. This is the good virus. We have libraries of tens of billions of human antibodies displayed on a bacterial virus or phage. And that's engineering at work. And to cut a long story short, we can now do in a test tube in weeks what the natural immune system takes many months. And crucially, we can do it in a way that's scalable. Once we get an antibody out of our system, as I'll show you, it has all these characteristics shown on the left. It's fully human, it's highly stable. It has a long half-life, it's potent, specific, low immunogenicity, identical to natural neutralizing antibodies. So it has all the characteristics you would put on a wish list for a therapeutic against a virus such as COVID-19. And this very process is a validated human therapeutic that's already been used for greater than 50 approved drugs. So we can now do this in real time, and I'll attempt to show you where we are and the speed we've gotten just over the last 2 months. And furthermore, this is a slide from our cancer research, which I mentioned was our main focus up until March. We had a whole decade of work. But the key point is we don't work in a vacuum because this is an in vitro system. The antibody platform shown in red is fully integrated with all of these other methods that we need to develop drugs. Cell lines, various assays. This is cancer, but it applies equally well, as I'll show you, with COVID-19. Once we come up with a new problem, we're not working in isolation. We can bring in many, many areas of modern biology. And the goal is then to rapidly take this platform, develop single molecules that are drug-like, and then move those forward very rapidly into animal models. Of course, the ultimate animal model is humans. So how have we done this previously? So this is about 10 years of data accumulated to track. We've targeted over 1,300 antigens and developed over 14,000 antibodies for cancer, some, as I'll show you, for infectious disease. But we're constantly doing, on average, 100 collaborative projects per year in our center. So this is not new to us. The process is well established. The only question is how rapidly can we mobilize the same methodology to a new threat such as COVID? So the key questions for us back in March were, can we apply the track pipeline to COVID-19? And if so, how quickly can we go from idea to drugs? So the answer to the first question was another great American. Yes, we can. Barack Obama said that about something else, but I think it applies to what we are trying to do. As I'll show you, we've been very rapid at this. And how quickly can we go from idea to drug? That one comes from me pretty damn quick. So that's where we are aiming to be. And now I'll actually get into a little bit of the real data, which is happening real time. I'll have to apologize if these slides aren't particularly polished. We're basically showing you what we're doing over the last month. But I think that's valuable, especially for a scientific crowd. So all of this started very, very quickly. and I'll attempt to show you at least some of what we've achieved in about the last 6 weeks. Before I do that, how can I say yes, we can when we haven't done it yet? We have a lot of evidence. That's what science works on. This is a new virus, but viruses aren't new. In fact, my group about 5 years ago worked during the Ebola outbreak, and we and others proved with published peer-reviewed literature with Ebola, which I must say in its effects has similarities to SARS CoV-1-2. It's an acute life cycle. It goes in quickly, and crucially, we've learned that if the immune system can mount an effective response quickly, then you recover and you remain resistant forever to that virus. So that's the saving grace of a virus like Ebola or SARS-1 or SARS-2. And what we've done about 5 years ago, we've proven as the outbreak was happening, but not in real time in that case, it was a retrospective study, we could make antibodies, make them fully human. And in mouse models, we showed something very important, which others were showing as well, so it's not just us. If you treat an Ebola-challenged mouse with this antibody that we developed in the lab, even after Ebola infection, you get close to 100% survival. Whereas Ebola is very lethal. In the absence of that antibody, you get 0 survival. And crucially, we showed something else important. If you take these surviving mice a month later, treat them with Ebola again, now nothing happens. So they become fully resistant. And that's a crucial thing. We think we can have a one and done therapy. You do one injection, our antibodies last for weeks. They start working with the natural immune system. The natural immune system then kicks in. The virus is cleared, you go back to life. Because the natural immune system is kicked in, you are safe essentially forever from that same virus. So we do believe not only would this be a treatment, but it would be a cure if we can administer it to people who have the virus. So this is post-infection, but in real time. So we and others have shown this. This is not new data. But there is new data coming out very encouragingly from ongoing plasma trials with the real deal COVID-19. So China's done this, Canada's now doing it, United States. And this is a rough but ready idea. If you have patients who have recovered, they have high titers of they have high titers of neutralizing antibodies. Why not take their plasma and inject that into patients who are symptomatic? And sure enough, it works. In the blood of every recovered patient, there is at least some neutralizing antibody, and it stays there for a few weeks. So if the timing is right, you take serum, you do a standard kind of blood transfusion with the plasma, and half a pint of plasma does give you therapy then in patients who are symptomatic but have not yet developed their own antibodies. So I think the data here is very encouraging. And for us, this is validating, then why not cut to the chase? If undefined variable plasma from patients is efficacious, why not make a drug, a single human antibody at scale, inject that, and hopefully get even better efficacy, but I would say definitely better predictability and safety, because you don't have to inject plasma from undefined source. Every person is different, and there's a risk of disease from other viruses, etc. And crucially, this is scalable. If we can make a milligram, we can make a gram, we can make a kilogram, we can make a ton. This has been done with other drugs. So this takes what we've learned from natural immunity and transfers it into an engineering universe. And that's basically what we started in my lab on March 24th. So everything I will show you actually started precisely 1 month ago. So it has been very rapid. So this is the real data. It looks a bit boring, but that's a good thing with data. It shouldn't be exciting. It should look like what it does. But very crucially, from March 24th, we started this. We got the antigen from a collaborator, Jim Reaney. We used our phage cycle. And within 5 days after that, we had this data. We had many antibodies that bind our target. RBD is the literal receptor binding domain of the virus. And crucially, they block binding of the host receptor. So in these two simple ELISAs, but they're simple yet powerful, we learned two important things. We now have in a test tube many different antibodies that recognize the tip of the viral spike. They recognize what we know is a neutralizing epitope. And literally that same day, Shane Mersch, who did these experiments at the bench, proved a subset of those also block binding of the host cell receptor. So these are prime candidates for disease therapeutics. And you can actually see the real numbers on the bottom. So within a week, we had 66 positive clones that bound and blocked the key interaction. A few days later, we had other data. To make a drug, it has to be more than just an ELISA. Again, this is real data. Sub-nanomolar binding to the virus-like particle. So this is actually a bigger version of the protein. This is representing the whole spike. And sub-nanomolar means therapeutically efficacious. That's the shorthand of medicinal chemistry and the science we're doing. So these not only bind, but they bind tightly enough. From a lot of other experiments, we know that they're tighter than actually natural human antibodies. So these are potent. Another way of measuring that is here. And crucially, at the very bottom, something simple and dull, we have the approved drug Herceptin, which has been used to treat tens of thousands of breast cancer patients. And then we have our protein at the bottom, the new antibody. And by many analytical methods, including chromatography, they're indistinguishable. So what we've achieved in about 2 weeks' time is made milligram quantities of a new IgG, binds potently to the virus, inhibits interaction with the host cell, and as far as production, etc., is identical to an already approved drug. So we have a process, the process for making Herceptin quite literally, that we can now apply at 1,000-liter scale, 10,000-liter scale, whatever scale we need to get the kilograms or even hundreds of kilograms that we need for therapeutics. So this was done by, I want to say, the first week of April. And this is another look at the real data of our 8 lead candidate clones happening in real time. So these are different columns. How tightly does it bind? How well behaved it is? Does it block the receptor binding? Is there good yield? Is it a molecule we could make as a drug? And we're very confident if any of these molecules work in the final assay we're now doing, which I'll describe, not only would we have an interesting scientific molecule, we would have a scalable drug molecule. And in fact, on April 20th, we shipped these antibodies to our collaborators at Washington University in St. Louis. So I want to stress this is based at Toronto, but we've already established a global network that I'll touch on, and collaborators in Washington University have been instrumental on this. I'm not mentioning their names because if all this fails, they shouldn't have to take responsibility, but it's a great group and they're clearly critical. And we already expect early next week to get the results back from the real deal live virus cell assay. So starting on March 24th, we expect by May 1st to know which of our antibodies actually inhibit the live virus with human host cells. I think that's very rapid by any measure, but it does rely on collaborations. And the project timeline is shown here. This is another raw data slide I use with my own group. Here's all the things we need to do on the left to get this into human trials. And here's all the things we have done or are doing. So in black is everything that we've done over the last few weeks. As I said, we're at this critical stage. We want to narrow it down to 2 lead compounds. We only need 1 in the end. And we hope by the end of April, so 6 weeks from when we started, I did have to decompress from my Rio vacation. So we really started on March 24th. That's when we got the funding and the material. But our goal is, there's a compression here I won't get into. This is in May and June, we're going to be working with regulatory authorities in Canada We've also had strong interest from Italy and India. We're contacting multiple governments. We see this as a team effort. We'd rather have more trials than less. And the response has been amazing. So we think, having spent the last month and a half getting the science done, June and July, May and June will be devoted to testing the drug in cells, scaling it up, and getting regulatory approval. July 1st is Canada Day, and we're setting that as our goal. So if we don't make that, our backup is July 4th. But we want to get this drug into people by July. It's an ambitious goal, but it's achievable based on our pipeline and the science. And if we can do that, then the scale up is a matter of a month or two after that. And we would then be relying on close cooperation with many governments, because the beauty of a drug like this is it is scalable. Once you have a cell line producing milligrams, because 50 approved drugs already use this technology, every major country has multiple production facilities that could be recruited to make kilograms and even tons of protein for rapid distribution. So we're quite excited about that. And as I said, we're already doing this science in Canada, working with NIH-funded work in St. Louis, and then moving on to Italy and India. So we see this already as a global collaboration that we established in April. So all of this happened really in this month that's still not closed out yet. So I wanted to finish with a philosophical slide. So if we can do all of this, what is stopping us from getting a safe, effective drug into people? And I think you have to look at institutions. Even science relies on institutions. And as we know, drug development is not only a scientific exercise. Many different institutions have to work together. And if those institutions don't work correctly, they create roadblocks. But I like to think every roadblock has the flip side, which is an opportunity. I'd rather go back to the yes, we can days than the no, we won't days. That's my one political statement for the day. I think we've learned that everything is possible, as Nelson Mandela said, when it happens. So the 5 institutions we need to make a safe, effective drug and get it to people, I'd say in order of increasing importance, are finance. You can't make anything without money. What we're doing is high tech, it requires money. And unfortunately, as soon as you introduce money into an occasion, you do get greed. That's human nature. And I'd say, working in drug development now, first at Genentech and now in academics for 20 years, what's been disappointing to me is that we now have come up with an idea that if a drug can't make at least $1 billion, it's not worth doing, which of course would be absurd in any other industry. Nobody says this is only worth $100 million. Yet this is the influence of Wall Street, I'll just say it. We rely on banking, we rely on finance, but not to make the crucial decisions that are go or no go. We should not be basing decisions on drug development on finance. It should come in at the front end and at the back end. But there's been a major misstep that many people now believe in pharma and even in my university, that money drives science. It does not. Money is a fuel, the car is basic scientific principles. But if you can correct that, the huge opportunity there is no shortage of money. Dr. Thangavula mentioned some major billionaires who apparently are very motivated to invest. We just need to convince people that investment will lead to good things down the road. So that's my comment on finance. You need people to invest early in things that seem small. The analogy I use here when I was at Genentech, Herceptin was the first drug approved, or second drug, Rituxan was the first humanized antibody. The market was zero in the year 2000. Now it's $150 billion annually. It's the fastest growing area of drug development. And that happened within a decade. We went from nothing. So even finance needs to understand, you got to get new ideas working to really see what their potential is. The other one is government, and I think in many ways we're seeing that right now. If this becomes political, it's a roadblock. If people want to decide who's to blame and who gets the credit, it leads nowhere. But you know, the United States has shown with the New Deal and other major initiatives, the Marshall Plan, there's a huge opportunity. There's no better mobilization engine than a government that's working effectively. So we do need governments to push for getting ideas from bench to bedside. And I don't think that's an unreasonable request at this time. The third is academics. I mean, I know this better than anybody, but bureaucracy is kind of the watchword in academics these days. We're so obsessed with getting people money to do what they already do. And I'm kind of proud that what we're doing now, we didn't do 2 months ago. Emergency funding kicked in, but it does frighten me a bit. I don't think I would have gotten the grants I've gotten due to the emergency environment if it was business as usual. I'm not a virologist. Why should I get virology money? But I think this has shown us clarity. When there's a big challenge, all good ideas eventually come from human minds. And that's where academics is the real engine of all innovation. So I'm hoping that we'll learn and not give up this emergency mentality, which quite frankly, I'm enjoying. Bureaucracy has been cut to zero. Every time I get an email from one of my bureaucratic groups, I just say COVID-19, and within days it's approved. So that's a good thing. I think we should stick with that. And then the science itself. I mean, scientists, despite what people might think, are trained to be conservative. We're supposed to work on what we work on, get better and better at that. But as we know, innovation is the opposite of that. And after 20 years of working on cancer, I'm actually enjoying working in a field which has always been a sideline to me. And I hope, you know, we'll maintain that idea that you want scientists who are very good at what they do, doing stuff they don't normally do. That's kind of the key point. And then, you know, it's Earth Day. You know, humanity at the end of the day drives everything. And I think the biggest roadblock right now is the lessons from this disease are leading to nationalism. The lesson is we shouldn't have let those people in. We shouldn't give them our data. We should not be thinking about what they can do for us, but what we can do for ourselves. And of course, that's a trap that we in kind of the global community know, but it's a real trap that I think could pose a significant threat to the drugs that we are developing not being made available to the global community. I'm not ashamed to say I believe in globalism. For every negative, there's endless positives. And I think COVID-19 will require kind of people acting locally. That's what we're doing. I literally can't leave my 1 square block. I walk to work. It's about as local as it gets, but I think the impact should be even more globally. And you know, I thank you for letting me have the opportunity to talk to you guys in at least in virtual space. Thank you.
Thank you, Dr. Sidhu. It's delightful to see your work, and we wish you all success in pushing this to fruition. All of the things you listed in the last slide are so poignant in the world today, and we'll have many more questions for you during our panel discussion. So stay ready and get your coffee ready. With that, we would like to go to our next speaker. And